Hello, it's a pleasure to be doing this for the Department of Bioengineering. My name is Siddharth Jhunjhunwala. I run a laboratory here in IISc in the Department of Bioengineering. My background is in biotechnology and bioengineering. I got my undergraduate degree in industrial biotechnology, a PhD in bioengineering, followed by a postdoctoral fellowship before I joined this department about 9.5 years ago. It's been a pleasure to be associated with this department, and we are trying to do things that are different from what many other departments in IISc do. Specifically, my lab is looking at how we can translate medical device biomaterial technologies into the clinic. And in that context, we look at how materials that go into the body as part of devices interact with many of the cells in the body. We are primarily interested in the immune cell interactions. And so a large part of my lab looks at how macrophage and neutrophils, which are specific types of immune cells, interact with these materials and how they change material properties, both physical as well as functional properties. And at the same time, how the medical device or the biomaterial that's implanted, how it affects the immune cell responses and the immune cell production and its immune cell activity. So currently in my laboratory, we have about 10 people, six PhD students, three staff members, and one undergraduate student who's spending time in the lab over the semester. These students work on varied projects, but with the main focus being immune cell biomaterial interaction. One of them, for example, works on this how neutrophils respond to implanted biomaterials in the context of type 2 diabetes. We have mouse models for studying these, and we have clinical collaborators who we work with to get samples from patients and understand how patient biology changes once they have diabetes and how that affects the interactions with biomaterials. Another student is looking at developing in vitro models to study the same without needing to do animal work. So, how do immune cells interact with 3D biomaterials in vitro? Another student is looking at how smaller materials, all the way down to the nanoscale, can be used to deliver small molecule therapeutics to immune cells to clear off bacterial infections or to change their activity and phenotype such that they can be now react differently in the context of diabetes, maybe not cause as much damage, which ends up happening in diabetic patients. I've had some students who also finished their PhD. We've had five students who have finished their PhD and defended, and now they're doing various things across the globe. Many of them are postdoctoral fellows, and one is currently in transitioning towards the industry.
Hi, I'm Sahana. I'm a second year PhD student in the immunoengineering lab. My PhD project mainly focuses on the increased severity of bacterial infections in diabetes, which is driven by chronic inflammation that changes the phenotype and functions of immune cells like neutrophils. Our lab mainly uses diabetic mice to perform in vivo infection studies and fibrosis studies, as well as human blood samples to perform immune cell profiling and functional assays. We have already developed a preclinical mouse model where there is a biomaterial implantation that causes alterations in neutrophils similar to those observed in the diabetic mice. Hence, this model can be used to study the increased immunopathology in conditions like diabetes. Overall, the lab works in the intersection of immunology, biomaterials, and inflammation studies with the aim of finding a cure to diseases linked with chronic inflammation.
Hello, I'm Abhinav Raghunath. I'm doing my second year PhD currently in Siddharth Jhunjhunwala's lab, and I work on primarily on diabetes and immunology and how diabetes alters the biochemical and other parameters in our body, and also how it affects the immunological cell functions like neutrophils and other alterations. And in our project, we have a longitudinal cohort that we are developing at IISc, which helps us in understanding how the diabetes progresses and how it affects other parameters, both blood parameters and immunological parameters. Our lab has a really great working atmosphere, and everyone is really friendly and helping each other.
Hi, I'm Prem Singh Anand. I'm working in Immunoengineering Lab, Department of Bioengineering, under Professor Siddharth Jhunjhunwala. I'm working on a disease called colitis, which happens in the intestine of humans. We try to mimic this colitis-like symptoms in mice models, where we use DSS, which is the compound for inducing this colitis symptom in mice. This colitis symptom causes disruptions in the epithelial layer of the intestine. Colitis by mean you can understand that it is the region of colon which is getting inflamed, that is why it is called colitis. So because we know that it is in the later part of the alimentary canal, treating this thing is very difficult. So people try to inject drugs in the peritoneum cavity to treat this disease, but these drugs can go anywhere, right? So what we are trying to do as a lab here, we are trying to get some drugs which could be delivered directly to the colon region. And how we are doing it? We are encapsulating it in different polymers so that it could reach to the colon region. But if anything you eat, it gets digested in the stomach. So we are trying to protect this drug so that it doesn't degrade in the stomach and reach the colon region. For that, we are using a strategy called polymeric coatings. So for each different section of alimentary canal, we'll have different types of coatings. So first coating will prevent the gastric degradation. Second coating will prevent or help in adhering to the intestine. And third layer will have a targeted system which will target just to the colitis region of the inflamed colon. So that's how we are trying to deliver our drug so that the drug can effectively work on the colitis by healing the inflammation. In the lab, we have a very chill environment. People are very friendly, especially Siddharth. He likes to play a lot with us. With all the interaction we have, it's generally fun. He used to do a lot of PGs in past few years, but he has stopped because we pulled his leg along. As a lab, I think everyone is multidisciplinary in the sense academic obviously, but everyone is talented in different things. Like some people play online gaming, some people do sports, some people in dancing. So we have a very huge different diversity in all these aspects, and it's a fun people. They all are amazing and fun people to interact with.
Yeah, hi, my name is Vinod. I'm a fifth year graduate student in Siddharth Jhunjhunwala's lab. So my work primarily focuses on profiling immune cells in chronic inflammatory conditions like diabetes, obesity, and maybe cancer. So what we try to do is we engineer biomaterials, or rather design biomaterials to profile these cells and changes that may occur. And we also try to modulate the immune cells using the same biomaterials loaded with any immunomodulatory drugs. My other objective of my study would be to create preclinical models to profile these immune cell changes that may occur during chronic inflammatory conditions, such that we could test multiple drugs at the in vivo setup. And our long-term focus would be to translate some of these drugs into the clinic and for a better therapeutic outcomes. And to know more about our lab, we are a fun lab we do a lot of activities outside academia. We do a lot of retreats, outing. Siddharth is a very nice person as a PI, so he's always approachable and he would give you many good constructive feedbacks, which will help your narrowing down your objectives and asking specific questions for your project. So please join our lab if you're interested and connect with us to get to know more about us. Thank you.
Hello everyone, this is Devangi Bhotto, and so I'm in my immunoengineering lab. I'm under the guidance of Professor Siddharth Jhunjhunwala. He's a very open-minded person and has fostered a lot of innovative ideas into me. So I'm basically working on tagging neutrophils. So in the market, currently we have seen that cancers are being detected by using PET, CT or MRI. But the problem with this is they cannot they cannot see or detect the cancers or the tumors which are deep-seated into our body. So in the literature, we have also seen that cancers or tumor formation is associated with inflammation. So what we have proposed is why not use neutrophils which extravasate preferentially into these inflammatory areas, which is even true for the tumor sites. So we are trying to tag the neutrophils with proper molecules, like for example, the photoacoustic molecules, so as to detect the tumors where they are going. So that detection will be where the neutrophils are actually migrating.
Hello, I'm Bhavana, a first year PhD in the immunoengineering lab of Dr. Siddharth Jhunjhunwala. The primary focus of this lab is immunoengineering, and my research goal is quite different from the lab's primary focus. I will be working on bacteria, which can be used for a process known as bio-cementation. This is a process which naturally occurs in some bacteria under the right conditions, where they are able to consolidate materials by producing calcium carbonate. I will be looking into how this bacteria's performance can be improved. I am looking into how the viability can be improved when the bacteria is placed in different conditions. I'm also looking into how this bacteria can further be used in Martian and lunar soil simulants and how it fares in different environments. The lab environment in Professor Siddharth Jhunjhunwala's lab is very welcoming and fosters a creative and collaborative environment. He also is a very supporting person and helps me find and refine my goals towards my PhD.